And then the next thing, if I want to figure out if it's a problem, how do I control it? Which of these nematodes do I decide to target my management strategy for? Now, when you guys send in samples, this is kind of what in our nematode database, what I see when I'm diagnosing these. And this is from 3 putting greens on one course that I received last year. And if you look here, they had 3 putting greens. In this one putting green, the one on the top, I had 227 root knot nematodes, 651 sting nematodes, and 6 stubby root nematodes. And the next one, there was 17 sting, 1,500 lance, and 8 stunt. And the bottom one, 346 sting, 675 lance, 407 stubby root, and 94 dagger nematodes. Okay, so basically, I'm looking at that top one. I mean, sting nematodes are off the chart. Okay, our threshold on ultradwarf Bermudas for high risk is 20. They got 651. Okay, and they're at moderate risk from root knot. In the middle one, I'm super high on lance and just moderate on sting. In the one on the bottom, they're very high on sting, very high on lance, very high on stubby root, and moderate on dagger nematodes. Okay, so which of these nematodes do I target, and are they a problem? Now, this is what's interesting about this particular golf course, because I visited this golf course, and this is what this this is the one on the top, okay, where I had 651 sting nematodes. I wasn't expecting to see any grass on the screen. I go down there, I'm looking at it like, wow, that looks pretty good. I mean, it doesn't look like it's got 651 sting nematodes to me. So all those nematodes in there, and the green still, to me, looked pretty good. Okay. Now, when I take a cup out of there and I look at this, my root system is about, or their root system, I should say, but the root system is about 1.5 inches deep here. Okay. And so now, ideally, we would like that root system to go down here a couple more inches, but it's about 1.5 inches deep. Okay. But I seem to have a lot of roots in that top 1.5 inches. That same week, I got samples from a golf course down the road from the first one. Okay, if you look here, my sting nematode counts on the top one, I had 7, which is barely in the moderate range. None in the second green, just moderate numbers in the third green. Okay, just barely into the high range on the fourth one, and just basically one green where they were really high. Okay. So I'm going to look at this top green up there where I had 7 sting nematodes. Okay, what does that green look like? Okay, this is what it looked like. Okay, from their putting green on that course. Okay, it looked really, really bad. Okay, but it only had 7 sting nematodes, which is barely into moderate. I would not expect it to look that bad if sting nematodes are the primary issue. Now, if we take our plug from there, this is what a cup looks like from that green. Okay, now the roots are probably no more deeper than on the other one, but there was a big difference in the way this is structured. I'll show you in a minute. But basically, I look at the putting green on the left, it has really high nematode counts, and the one on the right, which has low nematode counts. Okay, so that can be really confusing. Okay, what do these numbers really mean? Okay, how do we determine that? Are nematodes really the problem? So let's go through some problem solving. First thing I see here, like I said, the cups themselves, if you look at the root systems, they're about the same depth. Okay, about 1.5 inches worth of roots. Okay, but if you look at the cup on the left, see those nice clean edges. Okay, there's actually a considerable root mass here. Okay, so when I wash those roots off, you see here, I've got quite a bit of roots. They're not real deep, but I got a lot of them up in that top part. Now, if you look at the bottom, these roots coming out the bottom, I do see some nematode damage there, okay, which is characteristic of sting nematodes. So I do have some nematodes damaging, but they seem to be down below that 1.5 inch depth. So it's probably what's happening is they're controlling the nematodes well in the top part, but there's nematodes down here feeding and reproducing and increasing down here. Okay, which may explain why the green looked good, even despite having a lot of nematodes. So if you think about your nematodes here, if I've got, and again, this is my poor attempt at a cartoon. I'm not a graphic artist here. This is supposed to be grass with a lot of roots, okay? And a little red wiggle there is our nematodes. So if I've got just one nematode here, and it's feeding on those roots, it's probably not doing a whole lot. Okay, so I got, if I was sampling, I may not even find that one nematode there. So, but now it over time, it makes babies. And so now I've got, few nematodes here feeding on these roots. So if I was to sample, I may come back with low numbers. But if you look here, I've still got a good, healthy root system here, right? Even though there's some nematodes there, they're not causing a huge amount of damage at this point, okay? So as a sample right now, you come back at low numbers, but I got a good root system, but I may worry about these numbers increasing over time. Let's say that happens. Okay, now my nematodes are increasing, and now I've got a mess of nematodes here feeding on these roots, okay? If I was to sample right now, I've still got a lot of roots, but I've also have a lot of nematodes, okay? But now those nematodes are going to start damaging these roots. So now a few weeks later, a month later, I come in here, and now my root system is really compromised. I still have a mess of nematodes here. If I was to sample now, I've got high numbers of nematodes and very few roots. But now there's nothing for these nematodes to eat. So without anything to eat, these nematodes, these plant parasitic nematodes, have to feed on live plant tissue. Okay, so without roots, they start starving, and eventually now I got low numbers. Okay, now I've got few roots, low numbers of nematodes, and these few numbers of nematodes here are probably enough to keep any new roots from developing. Okay, so depending on when I sample, when I hit on this cycle, those numbers can mean very different things. Okay. So I'm going to, just for time, I'm going to skip some of this here. But basically, nematode information is a kind of a snapshot in time. It kind of tells you what's happening right now. But in a couple months, those numbers may be very different. They may be a lot higher. They may be a lot lower just 2 or 3 months later. Okay. Be aware that these, what comes back from a lab, and when we assign, like in my lab, we're going to assign for each type of nematode we find if you're at low, moderate, or high risk of damage from that nematode. These are just general guidelines, okay? And basically, without being able to look at the grass and look at the roots, which you can do and I can't, they can mean different things. So you got to use your judgment. And when the samples come back, look at your grass, look at your roots, and kind of make a determination on if nematodes are a problem or not, based upon what you're seeing in addition to what's coming back from the lab. Okay. And I'll talk about this here in a few minutes, but again, nematode counts are often not a good measurement of nematicide efficacy. One of the common questions I get, someone will put a nematode sample, or nematicide out, and then come back a month later and sample, and then they freak out because the numbers haven't went down. Okay, basically your best gauge if the nematicide is working is how well the turf and root are performing. Okay, are your root system getting better? Are your grass getting better? Nematode counts are not the best judge of if a nematicide is working. Here's a good example. This is from one of our nematicide trials. If you look at the root system on the left, okay, you see all that nice roots growing there. The one on the right, okay, you see a very compromised root system. Okay, now I've actually got higher nematode counts on that root system on the left than what I'm seeing on the right. Well, why is that? There's a lot more roots, there's a lot more food for those nematodes over on the left. So my treatment knocked those nematode populations down and allowed a good root system to develop. And now the same number of nematodes are going to be, the grass on the left can tolerate that same number of nematodes better than the root system on the right. Okay, so again, focus on your roots more than your nematode counts in those situations. Okay, so what are these nematode counts and these nematode assays useful for? Again, to help determine if nematodes are a problem, part of the problem, or even a potential problem. Because ideally, you guys want to sample before the grass gets bad. okay, so that you can keep the grass from getting bad. So if you come back in that moderate range, you say, I'm going to put my treatment out before a problem starts, okay? Also determine which nematodes to focus your treatment program on and to determine what to treat with, okay? Now, again, did the treatment work? Again, focus on your turf performance and root growth, not necessarily those nematode numbers. Otherwise, you're going to end up looking like this guy, because if you go chase the nematode numbers around, you're going to go crazy. Okay, so there's a lot of stressed out superintendents that are stressed out because they got high nematode counts, even though their grass looks really good. Okay, the second question we want to focus on is, does it matter what type of nematodes we're dealing with? Okay, and you probably gauged from some of the stuff we already talked about is yes, it does. Okay, because and so we need to know often what nematodes we have so that we can figure out what treatments we want to use. Okay, now back in the day here in Florida when we used a lot of Nemacure. Nemacure had both contact and systemic activity. It was a nerve toxin. It worked on all the types of nematodes we were dealing with. And so we didn't necessarily back in those days need to know what type of nematode we had. We just knew if we had a nematode problem, we used Nemacure. Okay. Well, our new chemistries coming out tend to be more selective. Okay. So we really don't have any good silver bullet for all the nematode issues that we're liable to face. Okay. Now you may just have one or two that one nematicide was really good for, but if you have another type of nematode, it may not work as well. Okay, so let's talk about sting nematode a little bit. Now, you guys in California, you have a few sting nematodes there in a few spots. This is a Class A quarantine pest in California, you know, it's not as common here in Florida where this nematode originates from. It's everywhere. Okay, so I deal with this nematode a lot, but probably very few of you guys ever have to manage sting nematodes. But what I wanted to talk about with sting nematode is this nematode is a nematode that is what we call an ectoparasite. It spends its whole life out in the soil. And so in the soil, we'll find eggs of this nematode, like you see in that box on the left. Well, in that little, actually, this is kind of true to size, that little one on the bottom there. And then what hatches out are second stage juvenile nematodes, which then become third stage juveniles and fourth stage juveniles, and finally adults. So if you see here, we got juveniles, we got adults, all come from the soil. This nematode spends its whole life out in the soil. You guys have a number of ectoparasitic nematodes in California. Like I said, a few of you guys have sting nematodes. But from what I see from California, the most common ectoparasitic nematodes you guys deal with are going to be spiral nematodes. Okay, and these tend to be can get really high on some of your cool season grasses there in California, and also ring nematodes and some of these other ones. But an ectoparasite like sting nematodes spends its whole life outside in the soil. And it has this structure here we call a stylet, which is like a little spear or needle. It pokes into the root, especially the root tips, and it sucks the cell contents out and kills the root meristem. So you typically on warm season grasses get an abbreviated stubbed up looking root system like this. Okay. Now on cool season grasses, it's not going to be symptomatic. Okay. On cool season grasses, you generally just get a general lack of roots. But on Bermuda grass and zoysia grass, you'll often get these stubbed up looking roots like this. Stubby roots is another one you guys have in California that can cause the same type of damage. Okay, and now in the work we do here in Florida, looking at this nematode, what we tend to find is the numbers of sting nematode tend to increase during the winter and spring, and typically peak in May, and then they go down in the summertime. This nematode drops down deeper in the soil. So what we found here in Florida with sting nematode, we need to time our applications generally in the cooler months, the fall and the spring, and not in the summer, because in the summer, this nematode goes down deeper in the soil where it's harder to get stuff down to it. Okay, so in October through May, sting nematode tends to be in the upper portion of the soil profile. In the summer, it drops down deeper, okay? And it's a lot easier to get treatments up top than it is to get them way down here, okay? So that's why we kind of target our treatment for this nematode during that time frame. Now, I will say that the population dynamics work on sting nematodes in California, they look like they behave differently than ours. So yours may behave differently. Okay, so that may not be the best time to treat for sting nematode in California. But here in the Southeast, in Florida, in Georgia, North Carolina, South Carolina, et cetera, we tend to target spring and fall. Okay, so for an ectoparasitic nematode like sting nematode, we want treatments that are going to be effective. They're going to either kill our nematode or paralyze or do something to it. Now, because these ectoparasites stay out in the soil, you can either get to them with a contact product that moves in soil solution and contacts the nematodes out in the soil, or something systemic that you can put on the grass that gets taken up and then inside the plant that the nematodes then take it up as they're feeding. Okay. To do that, now, basically, though, we don't have any currently, right at this moment, any good systemic nematicides available. Okay, so basically, we have all these contacts. So for a contact nematicide to get to these nematodes, it has to be mobile through thatch in water. So it's got to be water soluble and able to get through thatch to get down in the soil where these nematodes stay. Now, depending on where you are in California, I know it's different, and it's cooler up north. In southern, it'd be more similar to what we have here in Florida. But basically, in the warmer temperatures, your nematodes could be active year-round. Okay, and that being the case, you want something that you can either apply repeatedly or something that you can apply that's going to last for a long time. If you have something you can apply once that only lasts for a week, generally that's not going to give us as much control as we want. Okay, so basically for sting nematodes here in Florida, again, you guys don't have curfew. What you're left with, we found Indemnify to be very effective, providing long-term, slower acting control of sting nematode. We have found abamectin to be effective on sting nematode, but only about 3/4 of an inch to an inch deep, which most of those nematodes are below that. It doesn't get down to them. Okay. Zelto, which is a biological product, we've had kind of low activity. I don't consider it a standalone treatment, but it's something that we can rotate with something like Indemnify to kind of hold populations down if we treat it with something that tends to be more efficacious. Okay. Now, root knot nematodes, you guys have a lot of in California, as do we here in Florida. They're a problem in both warm and cool season grasses. So I want to spend a little bit of time talking about root knot nematodes because their life cycle is very different from sting nematodes. Okay, if you look at your top left panel, you should see a root with these like jelly-looking things sticking out of it. Those are root knot nematode egg masses. Okay, and inside this egg mass are these eggs. They can be hundreds of eggs inside this egg mass. And so if you look at the right, you see these eggs of these nematodes, and you see the young nematode curl up inside there. This is the second stage juvenile, and this is what hatches from this egg. If you go on the bottom left, you see these eggs have hatched. Now these second stage juveniles are coming out looking for a root. Now, once they find a root, and so they typically are going to enter in to a root tip. They go inside. If you look in the bottom right, this is a Bermuda grass root that these second stage juvenile nematodes have all gone inside. Okay, once they get inside there, they establish a permanent feeding site and then don't move anymore. They molt several times, go into a third and then fourth stage juvenile inside that root. You see now these have all been stained red, so you can see them inside. These are third stage juveniles. You see now the nematode starting to swell up. Okay, as it gets older, and it stays, but it can, after this point, it can't move anymore. It's locked into that spot, feeding on these feeding sites inside the root. Now, on the top right, you see we pulled a root back. You can see these nematodes all inside there. There's actually 30 nematodes right here, all in a bunch. Then they mature into an adults. Now, the female nematode here is even more swollen. You see she kind of looks like a grape or something there inside this root. You see her now starting to lay her eggs in this egg mass. What tends to happen, if you look on your bottom right, you see this nematode. Usually there's several of them in here. There actually are several in this gall that develops. They're not real big galls like you see on tomatoes or something, but small little discrete galls. There's several nematodes in here. And then the root, you see it's all starting to rot there where this nematode infection occurs. And so basically this root rots off right below where that nematode's infecting, okay? And so you get these, short roots that are rotted off here. And so as a consequence, over time, your nematode activity tends to be confined to the upper portion of the soil profile in this very shortened, abbreviated root system. So now what I'm telling you right now is what I know about warm season grasses. To date, no one has really studied the biology of your cool season root knot nematodes as well as what I have with warm season grasses. But basically on your Bermuda grasses and zoysia grasses, your root knot nematodes tend to be confined in roots growing in the thatch and in the extreme upper portion of the soil profile up here. So to control those root knot nematodes, unlike with sting nematode, these ectoparasites, I want something that's going to get bound up in the thatch, move slow, and not just wash on past where my nematodes are. Okay. Again, we don't have anything systemic. We're trying to use things with control these with contacts. Now, remember, this nematode spends most of its life inside the root. Okay. So with a contact product, the only time that it gets exposed to the pesticide is when that egg hatches, that little nematode comes out before it gets inside a root again, which can be sometimes minutes and sometimes hours and sometimes days. Okay. So another thing about root knot nematodes is they tend to prefer warmer temperatures. Generally, the hotter it is, the more active they are. And so we typically try to target our treatment program to the summer. Okay. And we are typically, because now abamectin, which there's 3 commercially available abamectin formulations with turf labels right now, abamectin binds up in thatch and doesn't move, okay, which is why it's not very effective on sting nematodes, for example. But because it tends to bind up there where the root knot nematode stays, it makes a good treatment for root knot nematodes, okay? Indemnify is very effective on root knot nematodes. Now, one thing I have found with root knot nematodes, though, is it's a little bit more mobile than abamectin. It kind of gets bind up in thatch, but it'll slowly work its way through and start moving down in the soil. At that point, now it's moved past where those root knot nematodes stay. And so then you want to put a second application on, okay? And if you're doing spot treatments rate, you could put a third and a fourth application, okay? Now you're getting really expensive. But basically, the more you can split up your indemnify applications, the better, okay? So if you're not using the spot treatment label, it's best to make 2 applications of the half rate as opposed to 1 application of the full rate. So again, so the more often I can apply this, the better it's going to work for those root knot nematodes. Again, curfew, you guys don't need to deal with there in California. Again, Zelto, something that has low activity that you can use if you're using something with indemnify to kind of help slow down the reinfestation. Okay, this is an ultra dwarf Bermuda grass putting green infested with root knot nematode, and you see that yellow blotching caused by these nematodes. And what I wanted to mention now is something we've added to our diagnostic lab in the past couple of years. I mentioned to you that we find that on warm season grasses, these root knot nematodes tend to be up in that extreme upper portion of the soil and up in the thatch. Well, our typical soil assay, we use what's called this Jenkins method, and we're extracting our nematodes from down here, okay? And this top part, we throw that away. So the root knot nematode juveniles we typically find down here in this soil are ones like on this bottom right. You see those little bumps all over it? It's being attacked by a parasite. And so usually we get our sickly nematodes that aren't very effective down deeper. Okay, so we found that soil extraction for warm season grasses doesn't give us a good diagnostic measure for root knot nematodes. Very good for sting nematodes and other nematodes, but not for root knot. So we went to extracting our nematodes using this mist extraction method where we take our samples, We want 4.5-inch diameter turf plugs with adhering roots. We wash the plugs off. We put them in this mist chamber. Our mister comes on for just 5 seconds every hour. And here's our 4 plugs with the thatch and adhering roots we put in here. Now, when our juvenile nematodes hatch from these roots, they get washed onto this coffee filter. They crawl through the coffee filter and they're dropped down in these flasks down here below. And so we have found this to be much more effective than soil extraction on warm season turf. Okay, so what you're seeing here, this is from a bunch of my nematicide trials where we did soil extraction and mist extraction. And what you're seeing, the blue are the counts we got from the soil, and the orange are the counts we got from our mist extraction. The bottom dotted line is our moderate threshold, and then the top line is our high risk threshold. And you see that we're much more inclined to come up in at a higher diagnosis using mist than soil extraction. Okay. Now, I want to look. Now, this is going to be pertinent to you guys in California specifically, because what we have found in our diagnostic lab, when we get samples that we're doing both soil and mist extraction with, not we're doing one or the other, but we've got samples, we're doing both, okay? If you look at our samples from Florida, where we basically all Bermuda grasses, you see here again that we have much higher recovery with mist than soil. In Texas, now for some reason, all the samples we get from Texas in the past couple of years have all been on Bermuda grass, okay? And again, you see very similar results. Now, for some reason, basically all the samples we get from California, except for one in the past 2 years, have been from cool season grasses. Okay, so we threw out the one we have Bermuda grass. So here you're looking only in cool season grasses, and you see here the exact opposite. We're seeing that we generally get much higher counts from the soil than from the mist, okay? And so it seems to be the opposite of what we find on a warm season grasses in Florida and Texas. Why might that be? Okay, I'm going to skip that for time's sake. Basically, in Florida, we have 2 species that we find on our warm season grasses, Melodigium graminis and Melodigium marylandi. In Texas, the same. However, in California, in surveys that were done by McClure and others in California and the Western US, they found that on Bermuda grasses, similar, we had the same 2 species we have in Florida and Texas. However, on the cool season grasses, they most commonly found these other 2 species. Okay, so what I'm getting at is these other 2 species may behave differently than what we find on these warm season grasses. Okay. Okay, I'm going to skip that. So basically,
Billy, I have a question on that real quick.
Yeah.
So if you're talking that you're not seeing it as much in the mist extraction and these are cool season samples in California, then and your sampling technique is further down and you're finding those juveniles further down, would that mean that they're going to be present further down longer, or they're all going to migrate back up into that thatch interface?
Okay, so again, no one has worked with these cool season root knot nematodes on cool season grasses as much as I have on these warm seasons. So I know a lot about warm season grasses and their root knot nematodes. What I can say is these tend to behave differently, okay? But we don't know as much about how they migrate in the soil like we do on these warm season grasses. So I just where I'm located, I just don't get a chance to work on those. And to date, no one has worked with them on the cool season grasses as much. So it's not known.
Gotcha.
Okay. So, yeah, so basically, if you got Bermuda grass in California, I would recommend, not necessarily every time, but you may want to have mist extraction done as well as soil extraction. However, if you have cool season grasses, probably just soil extraction is fine. Okay, I guess what I'm getting at. So you don't have to spend double doing 2 extraction methods if you got cool season grasses, probably. Now, I was talking to a superintendent recently that sends samples from California, and he seems to have the outlier where he gets higher counts from the mist and the soil, and he's doing, but so again, I can't say in every case in California, but that's kind of in general what we see. Okay. Now here's a nematode that we do not have at all in Florida, but you guys deal with a lot with in California, and that's Anguina pacifici, the Pacific shoot gall nematode. Okay. This is a juvenile from some samples we got from California this week. It's a really pretty nematode if you're into nematodes like I am. Okay. But this is a juvenile nematode. This is typically what we're going to be extracting from soil and also from plant tissue. Now, again, I don't work with this nematode, other than what we get coming in diagnostic samples. Okay, but from what is known from this nematode from others, basically what this nematode does is very different from all these other nematodes that live in soil and roots. This nematode tends to infest the crown tissue of POA, and it causes these galls or swellings here at the base here. Okay, look like little onions. All right, and these nematodes are going to be inside. Most of them are going to be inside of this plant. Okay, this is from some work that McClure did. He has since retired, but he was really doing a lot of work with these nematodes before he retired. He also did those surveys on root knot nematodes out there that I was referencing as well. But basically, if you took in the top left, here's the juveniles that have crawled up the outside of this grass. They've entered into the sheath area, okay, and they're crawling down into here. The next panel, panel B, in cross section, this is where now these juvenile nematodes have crawled down, and then they start eliciting this gall. Okay, if you see here in the middle, this developing gall, those are now female nematodes full of eggs inside here. They're laying their eggs. The gall gets bigger. You have more and more nematodes inside there. Okay. Now, basically, then when the grass, eventually that gall dies. Okay. And when it dies, the juvenile nematodes crawl out and go down in the soil. Okay. So again, similar to the root knot nematodes, what we find in a soil assay are going to be juveniles, or if we incubate these, they're going to be juveniles crawling out of these dying galls. Okay, the males, female. This is a species that reproduce sexually, males and females. They're all going to be inside this gall here. So if you rip the gall apart, is the only way you're going to find these adults. So it spends most of its time inside this plant. Okay, and if you guys, you're out in California, if you manage these, you've seen these. They cause these declining small areas that tend to coalesce and turn into large patches of dying POA. Okay, so again, I don't do nematicide trials on POA. On Anguina, but from what I've been reading from the work done by others out there in California, I'm going to reference now is basically, now ideally we want something systemic to control these nematodes, but we don't have systemic products. Okay, now because you'll find these nematodes down in the soil, but most of the activity, the infections occurring up in the upper soil profile and even in the crown tissue. Okay, so ideally we want something that we can apply repeatedly, or it's going to apply and last for a long time, and something that's going to stay up top, okay, not just wash on down past where our nematodes are. And what's interesting is there are some things that seem to work on this nematode that don't work well on other nematodes, okay? But what does work, okay, the research coming out of California has shown Indemnify to be highly effective on Pacific shoot gall nematode. Now, Indemnify has what we call it is systemic. It's in the xylem. Okay, so it can get taken up by roots and move up. Now, what I have not found it to be particularly effective systemically on root feeding nematodes. I don't know what kind of systemic effect it might have on these Pacific shoot gall nematodes up in the shoot. But it does seem to be more mobile in the plant than some other nematicides, even though it's not what I consider a true systemic. Okay, and so it tends to be highly effective. Abamectin, which has very limited translaminar movement, but it does seem to be helpful for these nematode as well. But you need to do multiple applications over time. And unlike with root nematodes, you don't want to wash it off the top right away, because it needs to you're trying to get these nematodes up in all that crown tissue and stuff. Okay, so again, it's different. And one thing that's different in California, they have had some limited efficacy from azithromycin products, which are these neem oil, which is a plant exudate. I have never had any success controlling nematodes in soil with this stuff, but they've had some suppression of Pacific shoot gall nematode. But again, probably not what you want to, it's best if you do it in rotation with these other chemistries. And I do recommend that you rotate these products, okay? Because again, with these new nematicides, the potential for resistance is higher than with older nematicide chemistries. And so, my recommendation is to rotate these things as much as possible. Okay, with that, it concludes my presentation. And I'm going to introduce my team. This is, as some of you mentioned, talking to Laban. That's Laban there with the beard. If you call the lab, he's the guy that's going to answer the phone. He is a very hardworking man right here. He's really been really good for us. And then Alex here manages the nematode diagnostic lab for me. He's the one that actually does most of the nematode counting. He's also a trained nematode taxonomist and helps us with species that does all the species identifications, working with us on new species descriptions. I have my Maria, that's my research lab manager, Chris, my research lab tech. And then I have various graduate students that help both in the assay lab and in my research program. If you want to ever get hold of me, here's my email address. You can follow me on Twitter. I'm at Nematode Guy. We also have a Facebook page. And then here's our Nematode Assay Lab webpage, where if you're going to send samples to our lab, this is where you go to get the forms, get information on how to send samples and all that kind of good stuff. And with that, I conclude. So thank you guys for inviting me. And if you have any questions, I will attempt to answer them.
Thanks, Billy. Appreciate it. Hey, I have a question. So you went through all the individual nematode numbers in terms of their thresholds, and we all know that thresholds are pretty loose at best. Mm-hmm. Do you ever add up all of the individual species to a grand total of nematodes and look at that number? I know Fred Warren, Fred at Michigan State, used to do that before he retired. Do you guys do that in the lab?
He would do that with spiral and ring nematodes. And I don't do that officially, but I do it unofficially. Okay, so basically, and here again, I mean, because I have enough ambiguity trying to figure out what type of nematode does. and sometimes actually if you got multiple nematodes, their effects can be additive. Sometimes they actually suppress each other. Okay, and so for example, spiral and sting nematodes, spiral nematodes tend to reduce the amount of damage from sting nematodes, believe it or not. So so I do not do that in the reports that I send out. However, when I'm advising people and talking to superintendents and they're asking me questions, we're kind of going through their more of a discussion process, then we can kind of do that at that point. Okay, you're moderate on several of these, even though you're not high on any of them, it still might be a good idea to go ahead and treat. You follow me? Yeah, I do that unofficially, but not officially.
Yeah, understand. So, and then when it comes to the mist extraction, now that you're figuring out that a mist extraction is better for root knot, are you changing those threshold numbers, modifying those, or
yeah,
still
I'm in the
sticking
process
with the
of doing
old
that.
stuff?
Yeah, so basically, right now I'm using the same thresholds for both methods, but next year they will be different. And so, yes, those are in the process of being modified currently. So what was interesting when we started regressing the numbers we've kind of from the soil and the roots, in most cases, there are no correlation, meaning you can have 0 in the soil and get 600 from the roots. You can hit 300 from the soil and get 500 from the roots. I mean, there's really no, it's not like you can, they really, for these warm season grasses, it's hard to really know anything about root knot nematodes on warm season grasses from that soil counts. Now, sometimes I can, if they're high, I can say you got a lot. Okay, you probably got even, so what you're going to see is a huge moderate range on these soil counts. Because basically, if all I have is soil counts, it is a very ambiguous guess on what those things actually mean. Okay, the miscounts on Bermuda grasses, I feel a lot better about what the numbers actually mean. So yeah, you're going to see a big, what moderate risk range for soil counts for root knot nematodes coming from me next year.
Okay, it's interesting. It's I think as you struggle with this, it's pretty clear that this is studying nematodes is a pain in the butt. That's the bottom line.
Now, I will mention. Now, I didn't talk to. We don't see as many lance nematodes out in California as you do here, but I will say, not sorry, bear people, but Indemnify has absolutely no efficacy on lance nematodes. Okay, so and also it doesn't seem to work very well on spiral nematodes either, which are closely related to lance nematodes. So again, I guess this again why it's important. What I was getting at to know what nematode I'm targeting on my treatment list with. And I cut out some things, but there's a whole process you have to go through figuring out if I get these different types of nematodes. I tend to have to figure out, okay, I'm going to target my treatments to one of those. So I got to figure which one's the most important one. And then I may not get some of these other ones. And so I may have to do something else for them. Or in some cases, there's really no effective treatments. Like lance nematodes, currently there's no effective treatments for those nematodes. And so again, I get, when people ask me, I'll get this call, we got nematodes, what nematicides should we use? Well, without knowing what nematodes you're dealing with, I can't answer that question. You really need to know what, which types of nematodes we're trying to manage here to be able to get to that, to advise what to treat with and when to treat it and all those types of things.
Billy, question for you. I'd seen some work done, and this was related to sting, but I wanted to see if you've done any further work with the correlation between something like Pythium and how those two correlate together with the overall disease activity.
Sure. So yes, one of my PhD students that finished here recently, her research was looking at interactions between Pythium and root knot and sting nematodes. Okay, and of course we're dealing with warm season grasses. Okay, this is on Bermuda grass, and basically she found that sting nematodes made less virulent Pythium more effective. Okay, so if you have, because you got all different types of Pythium, right? You got some that don't cause any disease at all, some that cause very little disease, some that are really bad. Okay, so the ones that were generally not problem Pythium or of low virulence, that are really not much of a problem, they made those worse. Okay, and then she found that but if you had really bad types of Pythium, it didn't make any difference. Now, what we found with root knot nematodes is the root knot nematodes actually made the grass less effective by the problem Pythium. And so the aggressive, virulent, disease-causing Pythiums actually were less effective if you had root knot nematodes there. So now with the mechanism we're not sure of. It may be that the nematodes are just triggering plant defenses that then defend the plant from Pythium. But that's kind of my hypothesis, but we don't know the reason why that is yet. But again, it gets back to this where, knowing what nematode you have and then to really understand how it's going to, affect, be related to Pythium, you have to know what type of Pythium you have too. And so, yeah, it gets, I tell you, the soil environment is so complicated. You got all these organisms, and it's difficult to study these things. It's not like you can shrink yourself down to a nematode size and go down and see what's going on. You got to try to get, do studies in unnatural systems to really study how they interact. And so and we find continually that these systems are very, very complicated.
How about any other soil-borne pathogens? You know of anybody doing any additional work besides Pythium?
We have, I without any actual research data to support this, but I from what I have observed continually over the years, I think that lance nematodes predispose grasses to getting warm season grasses to take all disease, the Gammanomyces, and also some Rhizoctonia root rot type pathogens as well. And also root knot nematodes, I think, interact a lot with those same pathogens as far as increasing the incidence of disease. But again, we have not looked at that comprehensively in research trials, but that's something that just me looking at stuff for 25 years that I kind of see a lot.
Got it.
All right. Any other questions out there? I didn't see any other questions in the chat. Okay, Billy, again, we appreciate your time today. And for those of you looking for California pesticide credits, please make sure your information is in the chat box, last name, first name, California pesticide license, and your email address. I will send you a quiz in the next 24 hours or so. And if you can complete that and turn that around pretty quickly, I'd certainly appreciate it. Those of you from Arizona, if we have anybody from Arizona, we need the same information, your Arizona pesticide license number, and just let us know that you're from Arizona. That way we don't send you the quiz from California. And if you're after GCSAA points, there is the code right there. Go to the gcsAA.org, the website, and fill out the education point affidavit. And if you have any questions, you can certainly contact me or Cam, and we can help you out with that. So with that, thanks again for joining us. We do have in 3 weeks, our next person speaking is Dr. Frank Wong. You all might remember him. Well, he spent quite a bit of time in California. He's going to give us an update on what's going on with the regulations for golf courses. And as you all know, Frank, he is always worth the price of admission. So that is on October 1st. And then October 22nd, Dr. Bill Kreuzer and myself finish it out with growth regulators. So again, we appreciate your time, and stay safe out there in California. Cam, any last words?
Nope. Thanks, everybody. Just a final reminder, the website es.bayer.us/sunshine series. That's where you can find the registration if you're not getting the emails from either My Bayer Rewards or the invites from myself. So make sure to pass that on. Thanks, guys.
Great. Thank you. Take care, guys. Bill, you still on? Dr. Crow? Dr. Crow? Okay. Thanks, Cam. We'll talk to you.